Continuing on in chapter 2, we're going to go over now all the components of the cell. So looking at the fluid we find inside, called the cytoplasm, and all of the different organelles. For this class, we're going to focus on the human cell, so a eukaryotic cell. In other classes like microbiology, you'll talk more about prokaryotic cells, but since our focus for this class is human anatomy, we're going to focus on what a human cell type would be, which is eukaryotic. So we're going to talk about the structure and functions of various organelles. We're going to talk about the endomembrane system, including the endoplasmic reticulum, Golgi apparatus, and lysosomes. And then we'll get into mitochondria and peroxisomes, and then get into what the cytoskeleton is capable of. There's three main parts, and then what do they do? So let's look at what we call the intracellular components. These are going to be all the parts of the cell that are on the inside. So we have the cytosol, which is like a gel or jelly-like substance that is inside of the cell. And this is the fluid that everything is like suspended in. And then organelles, which are like these membrane-bound specialized structures that perform a very specific function. And then the cytoplasm is the cytosol and the organelle. So it's all the internal components of the cell collectively. So if we're looking at a typical drawing of a human cell, this is again just a generic cell. It doesn't have any specialized features to it at this point. This is just very generic animal cell. We see all the different components. So we're just going to kind of identify at least a few of them. The ones I name you should definitely know, but the ones on this list, like definitely take the time to look at this picture, look at all of the components that are labeled, and at least have a rudimentary understanding of what they are and what they do. So first and foremost, we look at the plasma membrane. That would be the barrier, right? We've already talked about it to some degree already in the previous video, but creating that barrier between the inside and the outside, and it is a dynamic component in that it has some control over what comes in and what comes out, as well as the fact that it moves. It is not stationary. It's not a brick wall. Right, and all of the components of the plasma membrane can move around. Another big one would be the nucleus, right, where we hold our DNA, and the nucleolus inside there that helps make things like RNA, and then the chromatin, which is what the DNA and its associated proteins would be. Right here in this like bluish color on the outside of that nucleus, we have the endomembrane system in part. So this would be the rough as well as the smooth endoplasmic reticulum. And then coming off of it in pink would be the Golgi apparatus. So those three pieces together make up the endomembrane system. We have lysosomes and peroxisomes. We have mitochondria. We have the centrosome, which is composed of two centrioles, and then various filaments, intermediate filaments, microtubules, and microfilaments to make up the cytoskeleton. And so we're going to talk about each of these. So what is an organelle? What's the point of an organelle? It's so that we have some sort of location, some sort of structure that performs a specific function. And so we're going to talk about the three that make up the endomembrane system first. So these guys are going to work together when it comes to producing, packaging, and exporting cellular products. So you can think of them like factories and delivery systems in a way. And so the three parts are the endoplasmic reticulum, which we can break down into two pieces. We can break it down to smooth and rough, the Golgi apparatus, and lysosomes, which are coming from that Golgi apparatus. So let's start with the endoplasmic reticulum first, also called the ER for short. I'm going to continue calling it the ER. I'm going to shorten it to just ER since endoplasmic reticulum is a bit of a mouthful. And so there's two sides or two forms of the ER. There's the rough ER and the smooth ER, and they are named based on their appearance when you see them in like a micrograph. So the rough ER is called rough because it actually appears bumpy, and the reason it appears bumpy is because there's ribosomes actually stuck in the membrane of it. It's stuck along the edges. And so the rough ER's main function is to help produce and modify proteins. So ribosomes are really the ones making the proteins directly. Ribosomes are the machinery for that. But the rough ER is going to help with that process and help modify them. So the ribosomes that are stuck in the edge of it can actually produce those proteins and send them directly on the inside of the rough ER for modification. The smooth ER is mainly functioning in lipid synthesis, so creating fats, right? And then depending on the type of cell we're looking at, it can have some other specialized functions. So breaking down and metabolizing carbohydrates, it can break down certain toxins sometimes, and then one of the big things, and we'll see this function come back many times, if not in anatomy, definitely in physiology, is that the smooth ER can store and regulate calcium ions. We'll see calcium ions play a very important role in specific body functions and specific cellular functions, particularly when we're looking at things like muscle contraction. And so you want to keep that function in mind. Don't just look at lipid synthesis, although that is one of the big ones. You should definitely remember the smooth ER being involved in regulating and storing calcium ions. And so here's just a close-up, right, where we see the rough ER, and you see all those little pinkish dots that are on there. Those are each representing ribosomes. And so again, under a micrograph, right, obviously this is like a cartoon version of the endoplasmic reticulum, but if we were looking at a micrograph, we would still see those bumps, we would still see those studs on the rough ER. Whereas the smooth ER, there are no ribosomes because we're not involved in making proteins. And so we don't need the ribosomes there, so it has a very smooth appearance. But if you notice, they're kind of continuous with each other, right? They kind of flow from one into the other. The Golgi apparatus is then going to take a lot of the things that are produced in the ER and then modify them, sort them, and ship them off. So you'll hear analogies to saying Golgi apparatus like UPS or FedEx. I like to think of it like an Amazon fulfillment center, in that Amazon doesn't necessarily make all the products, right? UPS, FedEx, they don't make the products that they're shipping out, but they organize them. So if you think like a big warehouse, we can sort through those things, put some things in one box, other things in another box, and then send them to where they need to go. So that's how I describe the Golgi apparatus, like an Amazon fulfillment center that has a lot of stuff that was not made there, but gets sorted and packaged up to go to its destination. And the Golgi apparatus has two distinct sides to it, the cis face and the trans face. The cis face is where the Golgi is going to receive things, so we're going to receive products from like the rough ER. The trans face is where things leave, right? So things get transported away through the trans face. So if we look at all these together and see how they kind of work together, we have the rough ER here in blue with our little ribosomal studs in it. Something arrives from the rough ER to the cis face of our Golgi, and then it fuses, it dumps off its contents inside the Golgi. As they travel through, they get repackaged, sorted, put together in the assortment that they're needed to, and as they travel through, they get to the trans face, the trans end of it, where now we form a vesicle to secrete this out. And in this case, we're secreting something outside of the cell entirely, right? We've packaged it up and the vesicle has fused with the membrane, dumping this particular material on to the outside. But these vesicles that are being created by the Golgi can go all over the place. We can use them to secrete things outside of the cell. We can use them to move things around within the cell as well. So in terms of location, where the products are going, that can vary greatly. And then lysosomes. These guys are coming from the Golgi. They're produced by the Golgi, and these have specific enzymes in them to break down, digest things that are not needed anymore. So we can break down large components if there's some sort of large molecule that's been brought in, or let's say we have an old mitochondria that is just not functioning well, or it's been damaged. We can break down and recycle them. So I like to think of lysosomes as little recycling factories where we can take components of the cell that are either too big or worn out or damaged, break them down into their like root materials, and then use those root materials to build something else. We also see the lysosomes being involved in autophagy, which is self-eating, and autolysis, which is self-destruction, right? Because auto, you think self, lysis, to split or to break. And we see this happen in apoptosis. Now, hopefully you've heard the term apoptosis before. If you haven't, apoptosis is when a cell is programmed to self-destruct. It's programmed cell death. And it's a very controlled demolition of the cell. So sometimes a cell will die without that signal being sent, right? So something happens, something traumatic, right? An injury or something like that, and so cells are killed in that process, and it's very messy when that happens. But with apoptosis, this would be like the controlled demolition of a building. We don't need that building there anymore, but we also don't want to damage everything around the building, so it's a very controlled process, right? We're going to slowly break down that building, or make sure that when it collapses in on itself and doesn't break anything around it. So apoptosis is kind of that. It's that controlled demolition of a cell that's no longer needed. And this is really important. It's not a bad thing for a cell to die in the human body. In fact, the reason why your fingers are separate from each other is because of apoptosis that occurred in the tissue that used to connect all the digits together. Right? So if you didn't have apoptosis happen ever, you would have little mittens for hands instead of individual fingers. Moving on to energy production and detoxification, we get to the mitochondria and the peroxisomes. Now, most of the time we say mitochondria, and I'm included in this, we're talking about one. However, I will make a note that mitochondria is plural and the singular is mitochondrion, just in case you see mitochondrion. We're talking about the same thing there. But mitochondria play a very important role when it comes to creating our energy currency for the cells. Our energy currency, the molecule that we use to power things in the cells, is ATP, or adenosine triphosphate. And so the mitochondria, they use a process called cellular respiration to produce that ATP en masse. So you make a lot of ATP for us. We have ways to make ATP without using the mitochondria. However, they're not as efficient for us. And so we like to use cellular respiration in our cells. The mitochondria are responsible for that. Now, the mitochondria are a little weird in that they don't have one membrane, they have two membranes. So they have a double membrane. And the reason being that the thought is that mitochondria were actually free living at one point and then basically became part of our cells. And we're talking, this is some old stuff, so I won't get too heavy into that. But that's why there's a double membrane, is that the thought is that mitochondria used to live by themselves, and then a bigger cell took them in, they formed a symbiotic relationship, and now we're at the point where the mitochondria can't live on their own, and our cells can't live on their own either. Some other proof we have for that, or evidence I should say, for that is that mitochondria also have their own DNA that is separate from the DNA in the nucleus. So another little fun fact with the mitochondria. The peroxisomes, these guys are going to help with lipid metabolism and also detoxification. So they kind of work hand in hand with the smoothie R. We mentioned the smoothie R does some similar things here too. But one of the big things is with peroxisomes, when they have hydrogen atoms from various molecules, they transfer to oxygen to produce hydrogen peroxide, which then can be broken down into simple water. So we can basically prevent things from oxidizing. So let's take a look at the mitochondria. So it kind of looks like this little pill-like shape. So here is our cartoon drawing, which is nice and clean, and our micrograph on the right. And you can see how you can see those kind of inner folds. So we have kind of like a nice oval shape for the outer membrane, but the inner membrane folds in on itself, right? Like it's almost like deflating, right? And we're not going to get into the nitty-gritty of why that is, but those inner folds, that increased surface area, is pretty dang important for it to be able to do its cellular respiration process. And this is where that magic happens, right? Cell respiration will happen on the inside here, going across that membrane on the inside, so that we can produce massive amounts of ATP, which all of our cells need to help power lots and lots and lots of different reactions. And then here is our peroxisome, right? We have this crystalline core on the inside and then membrane on the outside. Now we'll get into the last piece, which is the cytoskeleton. The cytoskeleton Just like your own bony skeleton, right, in your body is there to provide support and help you move, the cytoskeleton has the same kind of function within one single cell. It's going to provide structure, right, and it's also going to allow for movement. The additional functions that the cytoskeleton has on top of that is also to help move things around. It can act almost like a highway system. And then to help the cell reproduce, to go from one cell to two. And there's three main components to the cytoskeleton: the microfilaments, the intermediate filaments, and the microtubules. So the microfilaments are made of tubulin. These guys are mainly to help give shape and structure to the cell so that the cell is not like this empty water balloon just kind of squished in on itself. It can help give a three-dimensional shape to our cell. And then the microfilaments is also where we have the centrioles. Right, so centrioles, these guys are going to serve as an origin point for microtubules. And these guys are going to be extremely important when it comes to the cell reproducing using mitosis. We will see these microtubules be used to make sure that the DNA, the chromosomes, end up on the right side of the cells when they're splitting. So, and then we'll talk a little bit about that, or hopefully you've already seen mitosis to some degree and have at least an idea of where I'm going with that. But basically, your cells, they've copied all of their DNA from start to finish. And then when we go to split the DNA apart so that we have one copy for the one cell and the other copy for the other cell, the microtubules and the centrioles are the ones that are really responsible for making sure that one copy is on one side and the other copy is placed on the other. That way we don't end up with extra information in either cell or missing information in either cell. So this is what these look like. So here are microtubules, right here, so you can see they're kind of a tubulin going around and around and around. Right here are our microfilaments, which are really, really thin. And then our intermediate filaments, these guys right here. All right, and then the one that we haven't talked about yet, the big one, the head honcho, the one in charge of everything, at least as far as we're talking, is the nucleus. And so we're going to talk about what the nucleus is. We're going to talk about some of the components and contents of the nucleus, including the nuclear membrane. And then we're going to talk about how DNA is organized within the nucleus. So the nucleus is the biggest of all the organelles and has a very important job of housing and protecting the DNA. Some cells, we looked at the picture, right, the generic one, and generally speaking, when you see that picture of a generic cell, you see one nucleus. And for the most part, that is true. Most cells have a single nucleus. However, there are actually some exceptions which may surprise you. Some cells actually have many nuclei, meaning they're multinucleated. And so your skeletal muscle fibers, your skeletal muscle cells are a really good example of that, where they have one single cell, but there's lots of nuclei on the inside. And actually that's one of the key things you can use to identify skeletal muscle cells. And then some cells actually have none. They don't have one. They're anucleated. It's not that they never had one, because you might be thinking, well, how do you even do things if you don't have a nucleus, if you don't have that DNA? And so red blood cells are an example for the anucleated cells. Red blood cells in their infancy do have a nucleus and organelles. However, as they mature, those organelles in that nucleus are ejected out. They're removed, basically, and so that they're left just kind of like empty, right? There's really not a whole lot going on in there. But red blood cells have a very specific job, right? They don't actually have to carry out much except just move hemoglobin, with oxygen around. And that's really all they have to do. And their lifespans are pretty short. You're looking at a couple, 3-4 months or so for the lifespan of a red blood cell. So some cells have more than one nucleus. Some cells don't have a nucleus. Most cells have one nucleus, if we're, trying to be honest there. Now the nucleus has its own membrane. It's again another double layer, right? It's a double membrane again. This is called the nuclear envelope, so it has a specific name. And going through these membranes, we have these little openings, these little holes that we call nuclear pores. And if you think about it, we don't want the entire DNA molecules to be like leaving the nucleus, but we do need to be able to move especially large things in and out of the nucleus. Things like proteins, RNA, solutes, those things need to be able to pass through this membrane, and so nuclear pores provide those openings for that. Inside of our nuclear envelope is the DNA. This DNA is organized into chromosomes. So if we're looking at a human, we're looking at 46 chromosomes found in 23 pairs. And that this DNA is condensed down in the form of chromatin. Chromatin is the DNA plus the proteins that help organize and regulate DNA, right? So it's a combination. DNA plus protein gives you chromatin, right? So why do we have those things? Because DNA is a massive molecule, which means we need to organize it. We need to regulate which parts of DNA are used and which parts are not. The proteins that are found with DNA in the form of chromatin help provide a scaffolding for the DNA to organize itself around, as well as proteins and markers that can help turn things on or off. So here is our nucleus, right? So you see that double membrane system around it. Here's all these little flower-like things, or the nuclear pores. You see how they go all the way through. We have the chromatin condensed down. So again, this is the DNA plus the proteins that help organize and regulate it. And then the nucleolus in the very middle here that almost looks like the egg yolk. That's going to be to help produce things like RNA. If we look at our DNA structure, this would be when it's very tightly packed if we're zoomed out, right? But if we zoom in, so we can start with our chromosome, we zoom in, we see our chromatin winding around. We see the DNA is in that blue wrapped around these gray circles. Those gray circles are histones and forming what's called a nucleosome. Again, these are those proteins that can help provide that organizing structure for DNA. And then the DNA itself in its helix shape. And then if we were to unwind it and look even closer, right, we would see the individual bases. And of course, this is a cartoon drawing of it, so it's not accurate in terms of how it actually physically looks, but it gives us that understanding.